Welcome, everyone, and thank you very much for joining us today for this new episode of our Discover Molecular Interactions webinar series. My name is Vivian, and I'm responsible for scientific communication at Dynamic Biosensors. And today we will talk about GPCRs and other transmembrane proteins. So each month in this webinar series, we will explore a different molecular interaction. So if you are also interested in other interactions, please check out our website. There are a lot of other episodes and recordings of previous versions. But now let's get started with the membrane targets. So transmembrane proteins, for example, GPCRs, have a very high therapeutic potential, and many drugs that are currently in use already target these membrane proteins. However, if we really want to tap into the full potential of these proteins, we need to characterize them, and this is often accompanied by a lot of challenges. For example, the proteins have transmembrane domains that actively influence oligomerization and also protein-protein interactions, and it is important to keep the native folding of these domains. And furthermore, these proteins are in a very complex lipid matrix, and this environment will also affect not only the structure, but also the function of the protein. And if we want to analyze transmembrane proteins, this is often done by isolating them, and these protocols are often very laborious, and we can also not make sure that we really keep the protein in its native state and the native folding. And this image of a cell is, of course, very simplified. And usually proteins are in a much more complex environment. We have a lot of cofactors, co-interactions with other receptors. We have post-translational modifications like glycosylations. So it is actually really obvious that the most relevant results we will get if we leave the protein just where it is in the cell membrane. So today I would like to present to you a technology that enables you to do just that and investigate the transmembrane protein directly on the cell. The technology is called Real-Time Interaction Cytometry, or short RTIC. And the instrument that is used to perform an RTIC measurement is displayed here on the left side. It's the Helix Cyto biosensor. And with RTIC, you can measure real-time kinetic rates like the association rates, the dissociation rates, but also affinities and avidities. The Helix Cyto is a chip-based biosensor, and on the right side, you can see the Helix Cyto chips, which have two measurement electrodes, one for referencing of background signal, for example, and one for your cell measurement. Now, the biggest challenge we had to overcome to be able to measure kinetics directly on cells was how to stably immobilize the cells on the surface of a chip. And our solution for this is shown here. You can see that we have these tiny flow permeable and biocompatible polymer cages, which we call cell traps. And the cell traps will now physically retain the cell on the chip surface. So this is very advantageous because you don't need to think about any specific surface molecules to capture and attach your cells to the surface. It is just a physical trapping of the cells in these little cages. I will now get to the workflow of an RTIC experiment, and it starts with this trapping of cells in these little cages. This is done automatically, so the autosampler will take up your cell sample and it will inject it into the microfluidic channel on the chip, and the cells will automatically be trapped in these cell traps. We will take snapshots of the chip surface so that you can control whether the cell capture was actually successful and how many cells you captured on the cell traps. In a second step, we will then measure the association. An RTIC experiment is based on fluorescence, so you need a fluorescently labeled analyte, and the analyte solution will then automatically be injected with stepwise increase of concentration, and by the increase in fluorescent signal, you can then derive the on rate of your interaction. This is followed by an injection of buffer, and this causes the fluorescently labeled analyte to dissociate again from the cell, so the fluorescent signal will decrease and you can derive the dissociation kinetics. And in the last step, we will then apply a reverse flow, and this will then push out the cell off the cell trap so that the chip is regenerated, the cell traps are empty again, and you can use it for the next cell experiment. These cell trap chips come in different variations. First of all, of course, different sizes to accommodate for different sizes of cells. So we have small, medium, and large sized cell traps on the chips, but we also have a different amount of cell traps on the chips, either only one trap if you're interested in the single cell resolution of your measurement, or you can also get them with 5 traps on the surface if you're interested in multi-cell measurements. These chips are also giving very reproducible results. So we tested this with the same interaction in either just a few measurements on a multi-trap chip or a lot of measurements on a single trap chip. And as you can see in this graph, the dissociation rates resulting from these measurements are very comparable. So now about the automated steps in the workflow. So I wanted to quickly show you what you have to do as a hands-on part in an RTIC measurement. And you can see here that there's only 3 steps which you have to do manually. So it's very simple. You just need to prepare your fluorescently labeled analyte. You harvest your cells. You can also fix your cells if that is something you're interested in. And then you place all your samples in the autosampler and start the assay, and everything else from the workflow will be automatically taken care of. So the hands-on time will result in approximately 30 minutes. So now I would like to show you some example data specifically for transmembrane targets. I have prepared 3 different case studies, which will all showcase a different transmembrane protein, but they will also demonstrate some useful features of this technology. The first example was a collaboration project with the Fraunhofer Institute for Cellular Therapies in Leipzig, and they are interested in DNA nanostructures, which could be used as platforms for therapeutic agents. So with these nanostructures, they wanted to change the valency of a specific analyte, and they wanted to see differences for different valencies in the interactions. So the model system they used are ephrine receptors on the cell surface, as well as ephrine mimicking peptides. And these DNA nanostructures would now contain either only 1 peptide or 2 peptides or 3 peptides on the nanostructures. So we have different valencies in our analyte, and we then tested the kinetics of these different nanostructures with their receptors on the cell surface. Now in this graph, you can see an example for a full kinetic measurement of the trivalent peptide structure. So we have 3 peptides binding to their receptors in this case. And this demonstrated how this multivalency really changed the affinity of the peptides for their receptor. Compared to values from literature for the single peptide, the affinity was enhanced from the high nanomolar to the low nanomolar range. And now with the trivalent situation, we have a stronger binding to the receptors, which is probably caused by the dimerization or trimerization of the receptors in this case. So you can see that we could derive the full K on, K off, and KD values from this interaction. But of course, we were also interested in the differences between the different valencies of these nanostructures. So we compared nanostructures containing no peptide, 1 peptide, 2 peptides, or 3 peptides. And these graphs now show you only the dissociation of the peptides. You see that for 3 peptides, we see the strong interaction that I've shown you before as a full kinetic, whereas for no 1 or 2 peptides, we don't see any binding. So we could clearly show the differences between mono and B versus trivalent binding to the cell surface. Now, we were very fortunate because our collaboration partners already published these results using flow cytometry to measure the binding affinities of the different constructs. So now we were able to compare our results to the flow cytometry results. And as you can see, we basically get the same result. That is that only the trivalent peptide binds to the surface receptors. However, if you take a look at the concentrations used for these experiments, we can see that we detected these differences at 150 times lower concentrations than the concentrations needed for the same result in a flow cytometry experiment. The second case study I would like to present is about GPCRs. GPCRs are a very important target in drug discovery, but there's still a lot of potential because they are very hard to analyze due to their low oxygen levels, and also because, as I said before, it's very difficult to express and purify them while still retaining their native folding. So we received 3 different cell lines from a collaboration partner, one with a very low expression level, one with a medium expression level, and one with a high expression level. And then we measured the binding of a specific antibody to these different cell lines. And in the left graph, you can see the total count of fluorescence we measured for each of these cell lines, the low, the medium, and the high expressing cells. Now, if we calculated the full fluorescence signal to a fluorescence per cell, we received this graph, which clearly shows the differences in expression levels between the high, medium, and low expressing cells. What is also very interesting is that our collaboration partner informed us that especially the low expressing cell line, which has a 12 times lower expression level compared to the high expressing cell line, was not even detected in a flow cytometry experiment. So this demonstrates that we can measure GPCRs with RTIC, and we also have a very high sensitivity, even if the GPCRs are expressed only to a very low level on the cell. Now, the final example are ion channels. Ion channels are also a nice target for new drug developments. They have a high therapeutic potential, but as for GPCRs, they are also difficult to handle. So we tried to measure them in our RTIC system, and we used a voltage-gated sodium channel as an example. This example also shows you how you could use a secondary labeling strategy instead of a directly labeled analyte. So in this case, we had a specific IgM against the sodium channel, as well as a labeled IgG against the IgM. In the left graph, you can see how we can measure the dissociation of the antibody from the ion channel. It binds very stably and only shows a very slow dissociation. And in the middle graph, you can now see that this binding is very specific, because in the middle graph, we used another cell line that does not express the ion channel. And in this case, we also don't see any fluorescent signal. Also in the right graph, we see another control measurement where we used only the secondary antibody without the primary specific antibody. And here you can also see no binding and no fluorescent signal. We repeated these dissociation measurements to show the reproducibility, and you can see that even with the secondary labeling strategy, you see very reproducible results of this biphasic dissociation. So in summary, these case studies showed you some examples what you could do with real-time interaction cytometry. The first example showed you how multivalency can affect the affinity of an interaction and how you can differentiate these different affinities using RTIC, and also that we use much lower analyte concentrations. And the second example showed you how to examine GPCRs, even if they are very lowly expressed, and how to also differentiate different cell lines with different expression levels. And in the final example, we were investigating ion channels as well as secondary labeling strategies, although we would always recommend, of course, to use the direct labeling strategies. So I hope that this could give you a quick overview of how you could investigate transmembrane proteins with an RTIC measurement. And if you have any specific ideas, any specific molecules you would like to test in the system, please always contact us, either me directly or through our website. And we can always arrange a demonstration using your specific samples for you to test whether it works in your case too. And just also a quick final remark. This technology is very new, so I know there's not much data on our website yet. However, we had a lot of collaborations and they will also show their data on our user meeting. So if you're interested to see more data and also discuss it with users who have already tried it, then please feel free to register for our user meeting, which takes place in September. So thank you all for your attention. And now I'm very happy to answer all your questions. You can use the chat function, the Q&A function, or you can also raise your hand. I can also unmute yourself. I'm happy to also directly talk about your interests. So the first question is how RTIC compares with SPR. Technology-wise, this is of course very different. I will quickly go back to the workflow, because I think that demonstrates it rather nicely. So you see that that we will collect the signal from the whole cell. So the analytes can bind to the cell, and we will measure the fluorescence coming from all the interactions around the cell. On an SPR measurement, you could, of course, also try to immobilize your cell on the chip surface. However, there are especially two major drawbacks, I would say. Like the first drawback is that this is often not stable enough. So if you want to apply higher flow rates, your cells are very likely to get lost. Whereas since we have these cages, the cells are much more stably immobilized and you do not depend on any attachment through surface molecules to the chip surface. And then the second, probably even more important difference is that for SPR, the depth of detection you can measure is very limited. It's only approximately 200 nanometers, whereas the cell is much bigger. So you only receive signals from the lower part of the cell that is attached to the chip surface in SPR, whereas in our system, we collect the signal from the whole channel. So we will see the whole cell and not only the small surface area. And then, of course, there's also the second part of the question. Concerning the weights, I guess. So there are some examples where we measured similar weights as with SPR. But there are also examples where we don't. And I think this is absolutely what we also want, because now we are measuring kinetics on cells, right? And we now want to detect all the co-interactions and everything that's going on the cell surface. So it would be very surprising if the RTIC measurements would always be exactly the same as a surface measurement on a biosensor, where you have the isolated proteins. And this would be very surprising. And then we also wouldn't really need to measure kinetics on cells, right? So we have examples where the rates are very, very similar, but we also have examples where they are not. And this actually gives you more information and shows you how the cell surface actually influences the kinetics. So I hope that answered your question. What is the assay volume and cell density? So the assay volume, the volume of cells you need, is usually 35 microliter for one run, with a concentration of cells of about 1 million cells per milliliter. So you need about 35,000 cells for one measurement. And concerning the expression level of the receptors on your cell, we have a sensitivity down to about 1,000 molecules per cell. Would that work for small molecules fluorescently labeled? Yes, definitely. We've already done so. If you have the strategy to label your small molecules, there's absolutely no reason that this shouldn't work. Would it be possible to establish a displacement assay? Yeah, sure. I mean, we don't have any preset assay for competition assays right now, but we are always happy to test this. And from the technology's perspective, this should work. So if you have an application for a competition assay or a displacement, yeah, I'm happy to discuss this in more detail, and we could just try it. But I don't see why this should not be possible. Okay, then we have a question if we have the system in the USA. We do. We have two We have a daughter company in the USA. So it's in Woburn, the Dynamic Biosensors Inc. We also have an office in Boston. So yeah, you can also go there for a demonstration or send your samples there if you are placed in the US. There's no problem with that. And then there's the question about the costs. I have to say I am not really a sales expert, so for the direct costs, I would redirect you to our sales representatives. They are happy to contact you and give you a specific quote. But I can definitely tell you that it is a very competitive pricing. If you compare it, for example, to state-of-the-art FACS experiments or BIACOR 8K, we will be in the same range or even below that price range. Is the relatively low number of cell traps limiting when cell populations show heterogeneity? And are chips with more cell traps available? So for the second part, for now, we have chips with 5 cell traps. That's the highest we can go. But the instrument has place for 5 chips. So you can do automatically several runs. You have 5 chips, which you can use automatically and exchange automatically. The low number of cell traps is usually not limiting so far. I don't know how heterogeneous your cell population is. There's definitely the limit that we don't know what we trap because the system will just wash the cells on the chip and we will trap whatever cell is there, and we cannot specifically say which cell is now in the trap and which cell is not. But if you use, for example, single trap chips, you could of course see the differences on a single cell resolution level, and then you would just need to measure more assays to see differences in the populations. Can you run a multiplexing assay with more than one fluorophore? Yeah, sure. You can insert plates into the autosampler. You can, for example, use a 96 well plate in the autosampler and just prepare different concentrations of different analytes in this well plate, and then just set up an assay which will automatically measure one analyte after the other. So yeah, you can, like, multiplexing at the same time is not possible at the moment, but one after the other is definitely possible. And you can also measure 2 different colors. Like at the moment, we are not measuring 2 colors at the same time, but you can label your analytes in either red or green fluorescent dyes. Both is possible. So if there are no more questions, of course, if you later on think about any other question that came up, please just shoot us a message or visit our website. There are also contact details there. I will put up my email address again. So yeah, I hope you enjoyed the webinar. Stay tuned on social media and on our website for future episodes of the webinar. I think the next one will also be very interesting about multi-domain RNA binders, if that's something you're curious about. And yeah, thank you all for your attention. Thank you for the discussions. I hope we answered all your questions, and have a nice rest of your day.